Hello, everyone. Welcome to the Human Cell Atlas Biological Network seminar. Today, we're delighted to provide you with updates on Atlas integration and cell annotation platform. We would like to remind you to join the HCA. You can navigate to our webpage. Look through the list of the bionetworks. We have 18. Please email us to join one or as many as you want. We are always happy to welcome new members. If you would like to submit your publication for inclusion in the HCA publication list, please also visit our webpage. It will show you the instruction. It will help your paper gain more visibility. We would like to remind you of the upcoming HCA events. The registration to these will open in the coming weeks. About a month from today, we have a Focus on Technology virtual webinar on March 9th. On April 27th, we are happy to welcome you to the HCA-wide town hall, also virtual, and please stay tuned for the registration for the HCA general meeting, hybrid meeting that will be held in Toronto in July. So please look out for the invitations in the coming weeks. Today's seminar agenda. The updates on Atlas integration will be presented by Malte Lücken. Then Evan Biederstedt will present cell annotation platform, and today's discussion will be moderated by Chloé Villani. With this, I would like to invite Chloé and Malte and Evan to come on screen briefly while I introduce them formally. So Chloé will be moderating today's discussions. She is the coordinating PI for the cell annotation platform. She's one of the members of the HCA organizing committee and one of the coordinators of the HCA Immune Bionetwork. Malte is a group leader at the Institutes for Lung Health and Immunity and the Institute of Computational Biology at Helmholtz, Munich. There, he leads the Integrated Genomics Lab, which uses machine learning and single-cell technologies to build population-level reference atlases and derive patient-level insights from these atlases that can be translated to clinical applications. The Lücken lab applies these approaches to the healthy lung and various diseases, including COVID-19, COPD, and interstitial lung disease. Malte is also co-founder of the living benchmarking project, Open Problems in Single-Cell Analysis, which aims to formalize tasks in single-cell data science and set quality standards for the evaluation of these tasks. At the Human Cell Atlas, Malte leads the integration team, focusing on building integrated single-cell reference atlases for HCA bionetworks, organs, and tissues. And Evan Biederstedt is a computational biologist at Harvard Medical School, now spearheading the development of the cell annotation platform, CAP, which is an infrastructure designed to improve our ability to interpret cellular state by facilitating the accumulation, sharing, and analysis of relevant metadata and molecular signatures. So thank you very much to all three of you for joining us today. And Malte, please take it away.
Thank you very much for the introduction, Ellen, and also for the opportunity to speak here today. As Ellen already mentioned, I'm involved in leading the HCA, or the newly founded HCA integration team, which I'd like to give you an update on the work today. If you're interested in anything I have to say, please don't hesitate to get in touch by email or various forms of social media that I have kind of pasted here below. As I'm sure, I don't need to introduce again, but single-cell atlases have kind of provided high-resolution cellular views into organs, which has led to kind of groundbreaking discoveries of new cellular identities, description of developmental processes, and cell states that are associated with disease or disease processes. With the kind of increasing commoditization and therefore accessibility of this technology, there's large research interests and many studies have been generated, but these studies may disagree. When choosing a particular study as a reference to build further hypotheses, which of these studies should we use? This is something the HCA integration team is looking to tackle together with the bionetworks from the Human Cell Atlas, is to build kind of reference atlases, which are consensus depictions of a particular tissue or organ across these different efforts. As I mentioned, here's just a list of lung data sets that have been published in recent years, all for different purposes. Some of these depict the healthy lung, others focus on particular lung diseases, but all of them can be repurposed to kind of as different views of healthy human lungs, as even the disease studies have control samples associated with them. While the data sets were designed to ask particular questions, they can be repurposed to ask questions across all of these studies. And this is something which I call aggregated queries. These aggregated queries can enable us to look at, for example, in a robust manner, which cell types express entry factors for viruses such as SARS-CoV-2, which we've done a couple of years ago. We can check to see whether a particular disease-associated states are specific to a disease, or even, specific or even found in healthy individuals, maybe with particular demographics. And we can look at how cellular profiles differ between individuals in numbers that any individual study may not be able to generate, given the time and the cost associated. And recently, there's been a great review in Nature Medicine from Aviv and Sarah's labs to kind of describing the longer term also implications, biomedical implications of such aggregated queries and integrated reference atlases, which are very diverse and lead from kind of understanding disease trajectories to using reference atlases and aggregated queries for drug screens and drug repurposing even. The difficulties with performing such aggregated queries per data set, rather than in all of them, and then aggregating the information per data set, is that it's unclear whether studies agree on the definition of cell types. Typically, there is disagreement in all different organs and tissues. In the lung, for example, there's a continuum between secretory cellular states, and the definition of what is a goblet cell and what is a club cell is not consistently defined in the same way across data sets. Any individual study typically represents a particular subset of a population, often middle-aged white males still, and thus aggregating the results of these queries per data set will mean that we're aggregating maybe quite different results on different populations. And one of the most central technical effects is that there is a kind of a residual latent technical effect of a batch effect on the transcriptome, just because these cells and these samples were handled separately from one another. Now, I'm sure I don't need to introduce this again, but just to give you a quick background for what we're dealing with here, if a patient comes into clinic 1 and we take a sample of this individual, and in a typical single cell analysis pipeline, we would map the data that we get from this individual into a low dimensional space, which hopefully is separated by cellular identities, so clustering can enable us to understand something about these individuals, this individual cells. If a second patient comes into a separate clinic, maybe recruited into a different study by a different lab, we want to perform an aggregated query, we need to map this to the same space. And typically, these individuals will often have their cells separated not only by their cell identity, but also by the batch in which they're handled. These batch effects can have various technical as well as biological reasons. These were 2 different individuals. They maybe processed differently, these cells. The samples were taken at different locations, potentially. What we'd really like to have happen to be able to perform an aggregated query across this data set without having to worry about these effects is for this data to be mapped on top of one another using data integration methods. Fortunately for us, there are many methods out there, and this is just 16 of some of the more well-used or widely used versions here. But because there are so many methods out there, there's also many different versions of what an integrated reference atlas should be, or this integration should be. So to start to address this, we previously looked at generating ground truth annotations for a whole host of over 80 different batches of cells. We pre-processed these in similar manners and divided them into 13 different tasks, used some of these integration methods to find quite different outputs. And this is just an example from one study in the human lung, which had 3 different data sets associated, and giving you quite different versions of an integrated reference for this particular data set. And so because we cannot look at UMAPs for everything, and the human eye is not really a particularly good evaluator of this, we developed 14 different metrics to assess how well batch effects are removed, but also to assess whether any biological variance is actually removed along with these batch effects when integration is performed. And using this, we could get a hierarchy or a performance ranking for which methods perform best to integrate diverse lung data sets, which we use to build a first version of a kind of integrated human reference atlas of the lung. Here we took 14 data sets. We harmonized cellular annotations across these data sets in a manual manner to ensure that we could assess whether there's disagreement between these studies, and also to use this to annotate the final object. We harmonized the metadata that were collected independently by different data generators so that we can assess the effect that this metadata has on the transcriptome. And we mapped the anatomical location that all these samples were taken into particular regions of the lung and airways. And with this optimized pipeline to kind of find which method works best, we could generate a version of an integrated lung cell atlas, which we found had broadly represent different compartments, but where individual labels from different groups, from different studies disagreed. This is why we reached out to the community in the Lung Bio Network to leverage the expertise that is there to annotate, re-annotate this entire embedding. And here specifically, the individuals I'm mentioning below, each gave their opinion on all of these clusters. And then in a consensus manner, we were able to harmonize those opinions into one reference atlas, where each cell type that is annotated here, or cellular identity, is supported by multiple samples, multiple data sets. And we're left with 61 cell identities. Now, with this reference, we were able to discover rare and novel cell identities that hadn't been described in the adult human lung. We could contextualize variants, these phenotype-associated variants from genome-wide association studies with particular cell types, generating kind of hypotheses for mechanism of action, and look at how gene modules or transcriptomic effects co-varied with the various demographic covariates, such as age, SNCs, smoking status, BMI, anatomical location as well. And we were able to show how one can use this integrated reference to map new data on top and immediately then project the labels from that reference to annotate the new data in a rapid manner, and also use the embedding that the new data is projected onto as a basis to analyze this new data set, thereby vastly speeding up the analysis process of a new data set in the slated lung. Now, from this human lung cell atlas, there were several aspects which we felt were kind of general features of how integrated reference atlases would need to be built. This included curating high quality data sets, selecting optimal methods for integration, and there are many different strategies out there for integration, using expertise across the community to annotate these cells. So this is not a representation of an individual lab's or individual person's opinion of what this organ or this tissue should be annotated as. And then different ways to use this. And so within the integration team, we have now set up a team across 4 different sites, actually, which I'll introduce in a sec. At the first stage, we thought we can expand, let's see how we can expand this cookbook, apply it and expand it to 6 different projects, from brain to blood, as you show here in stage 1. In stage 2 of the integration team, we're looking to expand once these kind of first prototype, first kind of pioneer projects are in a good stage, to expand this to further bionetworks to see whether we can leverage the knowledge we now have gained throughout these first 6, and to expand that further, and maybe to use some of these strategies. And then we're looking to address questions of more systemic nature, moving really towards this human cell atlas as a whole. Can we integrate immune cells from different organs? Understanding the basis of that organ reference already, can we look into a disease or development as well, and see, look at this temporal aspect there as well. To introduce the team, at the moment, so we have 11 different people on the HCA integration team. This team is still, still expanding as well in some parts. Chuan is working on the brain together with Caitlin from Helmholtz from Sanger, predominantly based. Peng from Sanger as well, working on kidney. I'm mentioning here only the members of the integration team, but there's of course also BioNetwork collaborators who are just as important parts of all these teams to make sure that these projects are happening. We have a project on the small and large intestine, led by MSKCC, Genentech, and with also participation of Helmholtz, the version 2 of the lung and eye, both from people at Helmholtz, and blood is a collaboration from the integration team side with Sanger and Helmholtz, but of course also on the bionetwork side from Chloé, who's here, from Harvard Med, Newcastle in Singapore, and the Genome Institute of Singapore. So just to give you an idea of how one of these projects is approaching building an integrated reference atlas, we can look at the immune bionetwork project specifically to build an integrated reference of blood. Now you may ask, blood is there's so many data sets, it's so well studied already, why choose blood? And there's a whole host of different reasons for why this is a really interesting first version of what an immune atlas may be. Because there's, the recent pandemic means that we have a lot of disease data as well. So we can really showcase how to integrate disease and healthy data. These data sets are very, very, some of them very high quality. There's very large scale of this. So we're looking at thousands of individuals, which again creates a challenge for the technical integration part of integration. And also this motivates kind of the involvement of specifically Sham Prabhakar and his lab for the genetic and ancestral diversity network to look at how really, human variation is represented on this level of blood transcriptomes, as well as several different reasons. The approach that the blood reference atlas is being built with is a general kind of principled pipeline moving from data selection and QC through to harmonizing these labels, in part motivated from how it was done for the human lung, a benchmark of integration methods or just comparison of how to integrate this, clustering, reannotation, and downstream analysis. Several of these steps have been done and others are still in progress, among which are a really important part is the collection of detailed metadata on both experimental design technical aspects as well as disease and demographics, which give us more biological information to really empower this downstream analysis later. So currently, the dataset selection in QC leaves us with cells from over 2,000 donors, approximately 2,000 donors with over 9 million cells from 14 studies, which had been already previously curated by the bionetwork. To enable us to compare different integration methods, we need to harmonize the labels now between these. And this is something that the team is doing based on cell ontology mappings, but also based on a tool which is developed at Genentech called Similarity, where we're looking to automate a little bit of this harmonization of labels, which was previously done manually for the human lung cell atlas. So to quickly outline how this is done, if you've heard the talk at HCA Vienna at the general meeting last year on this, I'm sure that was a lot more detailed than what I'm going to do now. But essentially, Similarity is a pre-trained deep learning model, which is used predominantly for cell type label prediction, which Graham, who is also part of the team, developed. Here, this is trained on large amounts of data and into kind of a batch-free space, which Graham and his team uses to predict labels for these cell types. Now, Graham will be the first to admit that these predicted labels are not perfect for every individual cell for every study. And this is why actually what we're doing for label harmonization is not to predict new labels and use that as a label as a harmonized label set, but instead we're using this similar batch-free embedding that Similarity projects the data to, rather than the predicted labels. And you can imagine this as follows. If I have this batch-free embedding from this large 7 million cell data set, which Graham trained on, and I have 2 cell clusters from 2 different data sets, I could project this onto this space. Now, in this case, even though I'm maybe not super confident at which labels I am projecting to these data sets based on where they map between this red and orange cluster, what I can say is that both of these clusters map to similar spaces in this reference, and therefore they are probably should have the same label even if they have different labels initially. And in that way, we can look to build a cell type hierarchy, a preliminary cell type hierarchy, and then map maybe different ways that particular cell types are annotated or different resolutions to one another across these data sets, and then use that with further manual curation to really make sure that this is done in an okay manner. But using in this automated manner to vastly speed up the initial process. Moving on, there's a lot of questions we have to answer for how to integrate these data. Are we performing adequate batch correction? Have we taken into account disease? Should we be doing this per lineage rather than across lineages? And which batch covariate is important to take, sample, donor, data set, or some combination of the three? And finally, we're looking to re-annotate this integrated object in annotation jamborees, which I'm sure Evan is going to be able to tell you a little bit more about the technology to do that later. This is kind of one instance of the pipeline that the integration team has developed as a whole for how to approach Atlas building. Now, this is not a rigid computational pipeline that goes from beginning to end, where you can push your data through and you get an output. These are rather steps, general steps that that we are traversing to ensure that we are being rigorous in how we find the best tailored integration strategy for each individual project. This is divided up here into kind of data and metadata curation as first steps, defining the scope of what we're actually targeting, moving towards a technical phase of harmonizing cell type labels, ensuring that the cell type labels are sufficient, and if they are not, in relabeling particular data sets with high resolution labels when they weren't available initially, which we called seed annotation, and then comparing different ways of integrating this data to preserve the labels that we previously harmonized. And finally, in a biological insight step, the data integration happens, and then we're looking for kind of annotating this and performing downstream analysis. The central step of this is this kind of comparison of different integration strategies, where we're really looking into single cell versus single nucleus, per lineage integration, disease, how we deal with disease, or whether we need to rely on the data. And the different projects are at different phases of these pipelines, starting from very early phase, where we're still looking to exactly define what is a useful scope for a brain integration project, to lung V1, which is finished and has recently been accepted in intramedicine, and then with kind of blood and retina being more mature towards the lower stages of this process. Now, as I mentioned, probably the core part of this is to understand how to best, at least to get to the downstream analysis, is how to best generate such an integrated reference embedding. And I've shown you how blood is approaching this by first cell level harmonization. Where we are past this already is in the retina to a certain extent. So this is just as an example to show you how in the to build a multimodal reference of the human retina, we looked into how to optimally integrate this data while performing some of these other steps at the same time. Together with the iBio network, specifically the lab of Ray Chen and two of his postdocs, Jin Li and Jin Wang, Ignacio Ibarra from the TICE lab, who's also in the integration team, has been generating an integrated reference of single nuclear and single cell data sets. We found out quite early that there's quite a strong batch effect between these data sets. And here I show you just a PCA embedding of just pseudo bulked whole data set representations. And we kind of quite clearly see single nuclear data sets clustering more similarly compared to single cell data sets, possibly also because of compositional differences. For example, we see that Miller glia cells and astrocytes are better captured on single cell RNA-seq, while amacrine cells have a higher proportion in single nuclear RNA-seq based on these donors. There's a reasonable number of donors and a very comparatively large number of cells at over 2 million. We are also looking at per lineage reintegration of some of these cells, which are very, very rare, but have a high diversity, and which may not be captured, which have shown to not be captured super well when we're doing integration of the full data sets, because most of the variance that is explained in this embedding is devoted to cells which are much more prominent. As well, there's also an attack aspect to this and the mapping of the data onto spatial data sets to look at kind of the spatial context. To optimize the strategy for single cell and single nuclear integration, there have been several different approaches that have kind of been proposed to do this, right? In our previous benchmark, we found that SCVI, one of the tasks for brain data integration had both single cell and single nuclear data, and we found that ScanVI or SCVI were both fairly good at this. In contrast, there's like a landmark paper for the human heart, the first human heart atlas from 2020, where they integrated single nuclear and single cell data because cardiomyocytes were rather difficult to get in single cell given their size. which they were profiled in single nuclear then, and scgen was used here to integrate these data sets quite successfully in this paper. Also, as a result of a recent jamboree run by CZI to test how to quickly integrate data sets at very short timeframes, the Joseph lab, or Adam Gayosa from the Joseph lab, built a kind of preliminary version of how one might be able to adapt SCVI to better integrate single cell and single nuclear data with a masking on the genes, which is motivated by approaches that the Sachiji lab took to perform differential expression between single cell and single nuclear to optimize the feature selection for this integration. And so we tested this as well, all of these different approaches in our benchmarking pipeline to find that while this batch approach from SCVI and ScanVI did improve on the base models, still scgen seems to be the best performing approach to integrate single cell and single nuclear data. And this is something we can superficially see on UMAPs to a certain extent as well, where these two modalities seem to be better integrated with scgen. Now, while this integration strategy is being optimized, the postdoc from Ray Chan's lab had been looking at re-embedding some of these cells to at least pre-annotate some of the more challenging to annotate cells. And here it took the single nuclear data set alone, given that we were still having challenges with integrating across these two suspension types, and integrated with scVI because there was no labels that we had for scgen or ScanVI, both of which require preliminary cell type labels just for these bipolar cells. And because not that much is known in human bipolar cells, he used similar cell types from macaque and found that actually the markers he got from macaque after cleaning this data a little bit for doublets very nicely distinguished bipolar cells into like on and off polarization, and showing these kind of very distinct clusters, which could be annotated in the context of this cross-species comparison. Now, having this step, the next approach is then to kind of take these annotations from the individual embedding, project them back onto the single cell and single nuclear integration to facilitate the annotation of those, and then use this large integration effort to seed an annotation jamboree. And here again, I want to, I'm sure Evan is going to tell you a little bit more about, how the ecosystem or how CAP fits into the ecosystem to perform these kind of decentralized annotations. And we're also looking into using cell by gene to help visualize integrated reference atlases and gain everyone's opinions to build consensus representations of this. Now, this is one way in which we're interacting with the ecosystem. We're trying to build infrastructure for also other projects than the two I've shown you, and you leverage as much of the ecosystem infrastructure as we can in these projects. Specifically, I've mentioned several times that we're benchmarking a lot of different approaches for integration, and we have servers both at Helmholtz and at CZ BioHub, which we are using to do this in a larger scale, given the masses of data that we are processing for this. I've mentioned Similarity as a way to perform semi-automated label harmonization across studies. We are also getting a lot of support from the DCP and the Lattice team, specifically from Cell by Gene. We're working with Cell by Gene to wrangle data sets and curate them and ensure metadata is consistently formatted to enable these projects. And where metadata is insufficient, even after this data wrangling, we have kind of developed simple scripts around Google Sheets to reach out to data generators via the bio networks to collect other metadata that is still shareable, so that we can really perform this kind of covariate analysis to see how the transcriptomes change, cellular transcriptomes change with different covariates. And this is just some of the problems that we are tackling. I don't really want to go through all of them, all related around different challenges in data integration across these 6 projects. So hopefully, when these 6 projects can conclude, it will be a lot smarter. At the moment, we're still very much in the weeds of things. I do want to highlight briefly the work on metadata harmonization and improving metadata completeness, which Ciro, who's shared between my lab and the Tice lab, has been working on, looking at kind of how the DCP and Cell by Gene platforms store metadata and which metadata categories we need in the HCA integration team which we have so far identified 60, which are important to us, and how much of these are shared between these different platforms. And when they are not there, really generating this automated way to query further metadata. With that, I'd like to summarize. We have I hope I've convinced you that integrated reference atlases serve as kind of a consensus, a representation of cellular tissue, which we can use as a basis to get input from a lot of different individuals in the community to really get smarter as a group, rather than projecting something from individual labs. The HCA integration team is looking to expand how we can generate these, starting with what worked for the human lifestyle atlas and expanding beyond this, because this is not a fit to every single tissue or organ out there. We're building centralized infrastructure to do this and leveraging the ecosystem where available. And then, of course, we're trying to speed up some aspects with through targeted method developments, like using automated cell type label harmonization. And I wanted to add here some of the very pretty pictures of two projects I unfortunately couldn't talk too much about, of different options for building an integrated brain or brain sub-region atlas, as well as a vision for HLCAV2 to really be able to infer ancestry and use this across a wider population in the lung. And something I'd like to finish with is we're also learning how these atlases can be used, and I'd love to discuss if anyone has further ideas, for things that they haven't seen, they think would be really, really useful, how to use these integrated reference atlases. And we'd love to test this with you as well. And with that, I have to acknowledge a lot of people. I've divided this up into BioNetwork counterpart and HCA integration team here, just for your perusal. But really, these two parties are working very closely together on all of these projects. I have to thank the senior advisors from HCA and support staff who's been supporting this project a lot. And of course, the HLCA, which I mentioned extensively at the beginning. Thank you very much.
Thank you so much, Malte, for giving you such brilliant overview of ongoing efforts. To make sure we stay on time, we'll keep questions for the end. And during the panel discussion, we already have 4 questions lined up. We're going to move on to Evan's presentation on the cell annotation platform.
Excellent. Thank you, Chloe. And thank you, Malte. That was an excellent talk, which gives me a nice segue to discuss some of our strategies here with dealing with inconsistencies we have with labels. And thank you for the introduction and the invitation for me to speak. I've given several very quick talks in the past year, but it's nice to have a bit more time to walk through what CAP is doing. Right, so within the next 30 minutes, I'm going to give basically the following overview, basically motivation to, and Malte actually already did a very good job, motivation to the cell annotation problem that we're having. An introduction really to the architecture that CAP has. We'll go through current features. I'll do a live demo. Then I'll give a brief statement about the upcoming features we have and finish up with some details about outreach, questions, and so forth. So, again, Malte already gave a great introduction to this. We're going through the process of the 6 initial bionetworks to make an initial human cell atlas. And in the past several years, the community here, the single cell community, has done an excellent job of showing the potential of single cell genomics. There's been amazing publications whereby usually there's this figure within the publication showing a UMAP with cell labels. And we all know how this process works. We've done the data analysis here. We've spent the hours going through the manual creation here to look at the prominent, differentially expressed genes, the molecular signatures associated with these clusters, and we go and then we start labeling them. For those who might not be, I'm sure everyone on the call is familiar with this, but HBB, you know that this is associated with a red blood cell. So this cluster is probably the identity of a red blood cell. You go throughout this process, and this worked really well for publications. But now that we're in the stage where we're trying to integrate all these data sets, we're noticing there's all these inconsistencies between cell labels, and it's mostly just I think we're now really starting to see the magnitude of the problem, that this sort of approach really can't scale, and it's really not consistent of how we're making these cell annotations, or how do we actually harmonize them. And so, if the goal of we're all gathered here to actually start building in an iterative process these precise and accurate atlases of all cells within organs and within organisms, we need some sort of approach to actually start investigating this in a bit more of consistent fashion. So I've given this slide previously. The problems we have here is we need some medium to actually start looking at these inconsistencies and try to resolve them a bit. I just talked about the issues here that we have, that individual research groups studying the same organ and the same systems, they'll have inconsistent terms between these groups. And yeah, we need a, should move my, well, I can't actually move the Zoom thing a bit, but the approach can't quite scale what we're doing now. So just to go through these problems very quickly, so we're on the same page. A medium to actually start seeing the inconsistencies of these cell labels. It really is at its heart, I think, an informatics issue. It'd be nice to have a way, some sort of platform to quickly look at the differences between the molecular signatures of how these cell labels are used. And obviously, even within research groups studying the same system, there's these inconsistencies. Also, we have, again, these inconsistent terms, even within similar plots, not to pick on particular plots here in publications, these are CAP-PIs. You'll see that, we all do this. Basically, there's abbreviations, free text terms. Oftentimes we see subclustering between cells that we know, or let's say our mesenchymal cells or luminal cells, but there's no way to actually come up with some sort of unique nomenclature for this besides just saying it's a luminal cell 1, luminal cell 2. Also, we have very broad cell types such as immune cells or chromatin cells. And then the other problem, of course, is that just can't scale. We all know the pain of doing this in a manual way. So that's sort of the problem that we have right now. And I'd like to introduce, well, give introductions before, but walk through what our solution with the cell annotation platform. Basically, this is infrastructure designed to start resolving some of these differences and come up with some consensus nomenclature that we can iteratively go through. So the next part of the talk is just to walk through sort of the give more of a theoretical sort of documentation view of what CAP looks like before I do a demo. CAP at its heart is meant to be a data repository to collect the molecular data and collect the metadata associated with these cell labels such that we can start to resolve the differences between them and build out a comprehensive human cell atlas. The way we design this is that because there are these sort of inherent inconsistencies of how people do cell annotations, we had a process whereby we want individual researchers to sign up, and so that we can record your annotations and then track them. There's a way that you can then browse the molecular data with the annotations and then create new annotations via the browser. And if this all goes well and we accumulate the data at the scales that we're thinking, we'll have at the end of this Wikipedia pages that sort of summarize what these cell types are and how they're defined, right? So basic user workflow for those sort of trying to understand CAP is that we upload HCA data sets. There's some way to generate cell annotations, either the cell annotations, the basic cell labels exist within the data sets, or they can be generated by investigating the molecular data, and then we publish this for the community to view. And of course, it's a data portal as well, so you can browse the cell annotations of others and look at molecular signatures, and then there should be a process to download the annotations such that we can use them locally. So just in terms of how did we try to organize all of this data, we have basically collections of data sets, which we call a workspace. We're familiar with this let's say with Geo or with GitHub, for instance. And there's a part of this in which you want to version this. We want this to be time-tracked, versioned, and associated with the DOI. And I'll explain why this is the case in a bit later. And this is a common question I have is this related to a save? And I'll address that. And then, of course, there's data sets with cell labels in the data sets, and then there's metadata associated with these cell labels such that we can investigate the differences between cell labels a bit. So another thing to mention with CAP is that there's this controlled data access. This is the first thing people ask is, can I work on these things privately before I share them to the public with a publication? Yes, that's the whole point of a save. So privately, you can actually start working on your cell annotations before you publish them. And this is the way we set this up, is that every user, when they sign up to CAP, they have a user role. This is very similar to Google Docs or to GitHub and other sort of data sharing type platforms, whereby you can either just view the documentation, you can actually have the privileges to edit it, or you're in charge of owning the collections. And obviously this will be useful for it was initially designed for individual research groups to start resolving annotations, but with the initial bionetworks, it should be useful as well. So to stress this idea of a publication, this is initially this idea of like a software version, or when you publish something on Geo, you should have this timestamp, it should have a DOI, it should correspond to a paper. This is normally how people think of these collections of data sets, is that you do the sequencing, there's a collection of data sets, and then you have a paper as publication associated with these. And then you should be able to download the annotations and the data sets in a standardized format. So to explain this timestamp thing a bit, again, it's because of how the process of how this analysis will do. At the moment, I think many of the cell annotations will be very broad for people, especially if they're connecting back to the cell ontology. And with more and more molecular data, and as we discover, and distinguish what are the novel cell types and how do we define these novel cell types, with more data, you should be able to go back and start revising those earlier annotations. So because of all this, you need some sort of way to track annotations and re-annotations, as Malte was describing. There's the initial annotation process, harmonization, and then re-annotations. So that is basically describing some sort of way to describe changes throughout time. And a workspace, again, is just a collection of data sets, which I can walk through. We have autocompletes associated with the metadata. So for users who are with the metadata, following sort of the DCP and cell by gene file formats, they can be, if they do have a free form string they'd like to use, they can connect that with the corresponding ontology ID as this is designed. So, and this is an overview of just it's very difficult to actually start looking at the differences of cell labels without any context. So there's metadata that we're asking users to provide with these cell labels. This is something that has been asked of me from CAP since the beginning. Is scientists would like to know, why did you provide that cell label for that cluster? Why did you do this? What is the evidence or the rationale for this? So there's a way to do this, and we've also slowly been developing what metadata is important, which I'll describe in a bit. That marker genes, what are the marker genes that these scientists used? This is an important thing for us to start collecting. Just to reiterate, CAP is at its heart a data repository meant to be collecting all of this metadata and then summarizing it for users to use. So this is, again, one way that, an important way that people can start to distinguish why it is that one research group would define same cell type in a completely different way if this was collected. And this is something that's currently not in production. We've been designing is basically, how do you view relationships of cell annotations within the same data set? And it seems like the community is using Sankey diagrams, which are a very nice way to see that these are the same cell IDs and cell labels, and how do they correspond with other sets of annotations. So the new UI we've been working on is basically how do you start working with the molecular data such that you can sort of explore, what are the annotations associated with this data set? How are they defined? Basically do the things that we do now in R and Python and cell by gene and other sort of tools to, explore the data in some sort of coherent way. And this, of course, will lead to, a way that you can actually do annotations via the browser. This is meant not only to be a very useful tool for the entire community and a nice thing to present, but it's also a way to start helping people come up with a standardized way of doing annotation such that we can collect metadata in a standardized way. Here's the demo. Okay, so it's tricky to see. I think I'm here. Okay. So this is a basic, server that we have to do development, about 50 minutes. So I mean, the basic workflow is you log in. I think I remember my password. I do. As I said, you want to have annotations to be associated with researchers. We're trying to move beyond this paradigm that you're just looking at papers and then looking at the data sets without this context. So if there are differences in terms of how research groups are defining these cell annotations, this seemed to be the best sort of software we could design for, you know exactly who did this. You have an author on your site. We can give sort of minor features here outside of my planned workflow, but you can go through and give, metadata and details about what you'd expect for your profile. So you give details about who you are. So I was playing around with this previously, but maybe I can start from scratch so you can see how this should actually work. So we go to your profile, and I described previously that there's a workspace. So this is an example of a workspace. Somewhat embarrassing, I named it foobar right before the talk to make sure that it was working. So I'll delete this. So I'm going to delete the workspace. And if I run out of time, I'll just start working with published data sets. So the idea is that as a computational biologist, you start creating this collection, and this should correspond to some sort of paper. So you'd want to say something like, oh, study of pancreatic cells and metastasis, et cetera, et cetera. You give some sort of description about what the study is. Again, this is very similar to Geo. This is like the abstract of a paper, and you can give a scientific journal DOI if it exists, a link to the raw data if it is, for instance, on Geo, contact names, et cetera. And so you create this workspace, and then you can start uploading your data sets. I'll give a caveat on all this about how this should in principle work with future data sets. Let's see, I think I had not on the desktop. I have enough time. We can do this. Not on the desktop. No. Data should be in. Let's go. Well, given limited time, I can just actually look at some data sets. You upload the data set, and you'd be able to investigate them. If you go to the CAP website now, you can actually see, there's an upload process, which is very quick and very snazzy. You can see there would be published data sets that you can look at. You can actually see these are the cell labels. This alone is sort of where we are at the moment without actually having the molecular data. So you should be able to open up and look at the molecular data. Molecular data, this is going to take some time. With many of these data portals have this issue to actually start loading the data on the browser. And in this case, there's really just one set of cell labels that have been curated by cell by gene to start looking at what are the cell labels within this data set. There's various features associated with this new UI. That we have, there's a zoom of the heat map. If you select, there's two types of ways to select this. If you select, let's say, these large amounts of cells, which are the progenitor cells, you should be able to see what are the top markers associated with these progenitor cells. At the moment, these are, close to 20,000 cells. You can look at the overdispersion values to see, this seems to be a fully, overdispersed gene. That should be interesting to take a look at. That will highlight the heat map, I believe. Oh, okay. So there's something, of course, associated with B cells. There's a way that you can actually start looking through the heat map and exploring this a bit. You can see, what cell you're looking at and what is the Z-score associated with the cell. Let's see. There's also a way that you could do, doing selections and actually doing differential expression. At the moment, I think the UI has been created to really do explorations of data sets that have already been annotated, but there's also the beginnings of allowing users to actually do annotations on the fly, as it were. So I'd have to be logged in. I have to own the data set to actually do this. And I'm trying to find I would upload the data set here. Let me try one more time. Okay, study of pancreatic cells. I do have 2 minutes. It's maybe not that big a deal. Let me open up. Well, this is one reason I should have. Anyway, you're also able to see sort of relationships between these cell annotations. I think at the moment there's only one set of relationships here. We have UIs to actually ask users to give us this associated metadata, synonyms, categories, marker genes, and so forth. And then again, you can look at the molecular data. And given I have 10 minutes left, I should quickly get back to the upcoming features for the CAP. So I'm going back to my slides. Okay, so as a quick demo, I didn't get through the workflow of uploading the data set simply because I cannot find the data set I wanted to upload on my folder. But just to go back to the problems of how this could actually solve things, a medium to actually explore how these molecular signatures differ. Now that you're able just to see the data sets and click them on portals, you should be able to explore these genes. This exists on other sites as well. But the point of CAP was actually to be collecting metadata that's associated with these cell labels. So you can actually start resolving the differences between cell labels and then harmonizing them. And so, the main takeaway I had from the introduction is that the practice that we have now of just giving cell labels and then in the data set is not really sufficient to actually start integrating them. And there's several approaches that people are trying right now. You use free text labels, which I think people immediately saw this is not going to work given those first slides I showed. Then there's this idea that you would take a free text label and you would associate with an ontology ID, which I think what Malte was describing. This usually works in that if you can connect the free label term to an ontology ID, you then have standardized metadata, and then you can start integrating in a way which is sort of informatically sane. The issue with this is that usually you'll have a very broad term associated with the cell ontology. Just logically, if you have novel cell types that don't exist in the cell ontology, you'd have to map back those novel cell types to an existing cell ontology. And so there's sort of a loop here whereby it's not quite clear how do you add new terms to the cell ontology. I think the current way is that if you are a researcher and you have a new cell term that doesn't exist in the cell ontology, you should ask the cell ontology to add it. And this is actually why the cell ontology is working with CAP. This is David Sumi-Sutherland. So there's actually a lot of metadata that you need to solve these sorts of problems. Otherwise, it really is just doing what I described. You'd go back to the nearest broad term and somehow try to add terms underneath this. And it's actually not clear how this would scale for the entire community in a sort of consistent way. So we've been struggling with this for a while about basically what information would you need to do this, to actually start creating a comprehensive cell ontology. And you would need, there's cell ontology editors on the project that have been looking at to how you would do these sorts of things. So this is related to a cell ontology schema proposal, which is very similar to what I described. You should have a free text, you should have a cell ontology term, but you should also ask the scientists to explain how they define those cell labels, which is currently not captured within data sets at the moment. So marker genes, evidence, is this a broad term or a precise term? And of course, the issues that we're having now is, was this a manually curated label, or am I looking at this file just as is this an output of an algorithm? So all of this can be stored in specific metadata that we can then start providing to the community. And problem number 3 is basically how do we work with these automated, how do we get past this process of what we're doing now, where we do manual annotations, which is very time consuming and is the sort of a bane of many people's existence on this call. How do we actually start streamlining this process a bit such that we can do cell annotations in a far more quicker manner? And this gets into the upcoming features of these annotation predictions. So there's a basic workflow here. If you saw the previous, well, of course you did see the previous UI, you're on the call. If you think about the previous UI, whereby you're able to look at the UMAP and the molecular data and then add or subtract cell labels associated with that data set, there's something analogous here for cell type predictions. So with the cell type predictions, this should be able to give you some hints when you upload the data set about what the identity of these cell types are. And so this is a basic, not a basic, but this is a plot from the OSIF lab, the postdoc John Ergan Bear, basically showing an output of POP-V, which shows, I'll explain POP-V in a bit, but it shows like the confidence of these automated predictions. And this would be within the CAP workflow. If it's a highly predicted cell type, you can possibly trust that a bit more and then accept those predictions. But for the low confidence, you'd probably want to investigate these a bit and then use the web browsing the tool to do manual annotations via CAP. So this is an illustration from Gary Bader that's explaining annotation transfer. This is the idea that you have a reference data set and a query against that reference, sort of transfer the cell annotations from the reference to the query. The workflow for CAP is that the user would choose what sort of annotation transfer method that they would like. They would then actually have these annotations shown on this UI that I just presented, and users would then do the workflow I just described. They'd accept, edit, or decline those suggestions, and then be able to make them more precise or change them as they wish. And then of course they would do the CAP workflow that I just described. As I mentioned, this is from POP-V, is one of these annotation transfer methods that we're now working to integrate into CAP. The idea here is very clever. This is the idea that you would take multiple methods that have been developed in the past several years by groups to come up with these predictions. And what you would do is then you would use an ensemble of these predictions to see if there's agreement between these various cell prediction approaches. If there's agreement, you can say that there's probably some sort of higher likelihood that that is truly an accurate cell type, or at least maybe it's broad enough, the cell type, such that you would accept that immediately as a user. And for the where there's disagreement between these methods, you should be able to see that there's this is something that will require more manual annotation. And this is precisely what I just described. These are three methods. I believe this is Allen Brain Atlas data, whereby you'd look at the various predictions of these algorithms, and then you'd look to see what is the ensemble sort of confidence together, right? And you'd be able to see that because there's one, I think it's ScanV, there's a bit of a, I can't have a cursor, this is unfortunate, but there's a little bit of green by one of the clusters where it's slightly unclear if that's actually an accurate cell type prediction. When you use the ensemble together, you can see that it shows that there's, this is maybe something you should investigate as a user if you're doing manual annotation. And then we get to the real, maybe crown jewel of CAP is this idea that what are we really doing? We're collecting all of this metadata and we're collecting all of this molecular data. This is really an excellent training set to do machine learning in the future, right? So you'd have a training set that users being you, the community has given, and you'd be able to then predict what would annotations look like completely de novo if you were to upload a data set that is not annotated at all, right? So this is a different workflow. You have a data set with no annotations whatsoever. If there's enough data collected on CAP, and if the models are accurate enough, you should be able to just upload the data set and you'd have a prediction which is reliable enough that this would save so much time. And you can just start clicking and accepting or denying these accepting or rejecting these annotations. So this is work that's being spearheaded by Felix Fischer and David Fischer in the TICE lab. Basically, what we're prototyping now is you do iterated training on all the available data that we have. And the current model, which is what we show here, what Felix shows here, I should say, not me, is it does accurately predict sort of broad cell types based on unseen data, right? And the improvements that we're working on now is to make this more granular with more granular cell labels. And you can see this this NA is describing what I described previously. NA right now within this plot is high uncertainty. We don't know what the cell type is based on the predictions. So this would be a case whereby with the browser, you should be able to investigate these low certainty prediction cells just by using the various selection tools that I walked through. So this is the current state of CAP. If I have more time with the questions, I'll open up the website again and show you features that might be useful. It's a data repository at heart, as I say. We're trying to collect the metadata that would allow you, the community, to actually start resolving these differences between cell annotations and do what people would really like, which is harmonize them and then have an integrated result that we can share with the world to great fanfare. And then, of course, you have the, there's basically this upload process, which I didn't get to show, unfortunately, but I'll show this maybe in the later YouTube video. And a publication process. You saw the publications. There's a UI that, of course, you work with to work with the annotations and with the molecular data. Okay, so upcoming, as I said, there's annotation predictions. I think I showed you work from the Nir Yosef lab. I think Sarah Teichmann's models, cell typist were there. And, you see models from Fabian Theis as well. I'm very open that I want people to come email me and to offer new models, and then we can start working on integrating these within CAP. There's completely different architectural requirements for doing annotation transfer versus just de novo predictions. So this will be up and coming. As I say, I think the UI we have at the moment is really useful for exploring cell types and cell annotations. And I can go through actually how you could use it currently to actually select cells and rename them and create new sets of cell annotations and new sets of cell labels. But there are a few key things I think that we should improve here to do truly de novo manual annotation, as you do now with R and Python. And so we'll be working on this. been speaking to bionetworks for the past several months, I think, about ways that we could actually curate, not curate, to tailor basically the workflows and the UIs that we have for CAP for the individual sort of biological problems that you're working with. And I think there are some ways, given how I've seen how the individual sort of bionetworks are trying to come up with consensus for these annotations, that we have a plan going forward. And as I say, if this all goes well and we do collect the metadata and all the data sets that we'd like, we should be able to have these summary cards up and coming. And then we're doing outreach activities as well. Ellen Todres, who introduced the talk, and of course the boss Chloe, they are doing, well, actually I'm doing the user interviews with bionetworks. I think there's some email I have to respond to. There's some of you on this call. We're doing seminars, of course, so I can actually, not in such a high pressure environment, give a proper tutorial of CAP on YouTube, and then I'll show you how that works properly, so you can upload the data set. And then, of course, there's annotation jamborees, which Malte was mentioning. So, and as I say this, I'm finishing up. This whole process really is meant to be a community service, a community service for everyone on the call and with the HCA community. So it would be best if, not be best, participation in the sense of having feature requests and telling us what you'd like to see with the product is useful. What I want to avoid is this people quietly grumbling and so forth. So do email me, and we can have discussions about how to best improve this. So with that, I'd like to thank the PIs for the project. They're not in order of the list I gave, but at the top you see David Osumi-Sutherland, David Regev, then we have Chloe, at the bottom you see Fabian Theis, Niels Galenborg, John Marioni, and then we have Peter. And then there's special thank you for the people who've been, I very briefly, given the time, went through the work that we're doing. There's John Nickenberg and Nir Yosef have been really excellent with this project and helping out. And then we have the Fabian Theis lab, it's Felix Fischer and David Fischer, no relation. And then Ellen, of course, works with the project a good deal as well, and she's great in trying to keep me organized. And there's many others that we've been having discussions with as well to improve the workflows. And then, of course, the team I work with, I'm very grateful for the passionate amount of software developers that we have here. So every day is a joy working with them. So I really want to give a special thanks to people like Dionys Silguzin, Maxim Svetlakov, Nick Akhmetov, Levan, Misha, Sultan, Tigran, Kostya, Ur, Pavel, Denis, Andre, and others. So thanks.
Yeah. Thank you, Evan, for this great overview of the cell annotation platforms. And for those of you who would like to get a detailed demonstration on how to navigate CAP at a slower pace and also, upload your data set de novo, we'll be hosting workshops, so you'll get that opportunity. So thank you, Evan. This was great. I'll also invite Malte to come back and we'll have some discussions, lots of questions in the Q&A. And Malte, I see you've been hard at work answering some of them already. So thank you for doing so. Maybe there's a question that you have answered, but I'm not sure that everybody saw. And there's another question of the same theme that I'm going to ask because Evan can also jump in, which is from an anonymous attendee. What is the difference between the cell label harmonization phase and the downstream cell annotation phase? Are the harmonized label reassessed in the annotation process? And I think perhaps some of the audience may confuse the process of the first phase of the integrations for the batch correction versus actually annotating the atlas. So perhaps you want to go first, Malte, and then I'll have Evan add.
Yeah, that's perfect. Thanks. It's a really good question. I probably should have been more clear on the difference between these two steps. So cell label harmonization, we are not attempting to re-annotate any data set. The purpose of this is to make sure that the annotations, I think Evan also showed, how there's disagreements in these kind of labels that are given very often, and how he was showing the cell ontology helps with this, with these kind of different nomenclatures. And here we're looking to take the clusters that were originally annotated in each data set and just match those clusters to one another rather than re-annotating each cell. What we've previously found, we did this re-annotation effort later. So in the first, the label harmonization is just to say, like, okay, is B the same as B cells by someone else, right? A computer cannot match those two. It can be as simple as that. And this we need to assess based on the original labels on a same nomenclature, with like how integration methods perform differently. In the re-annotation effort, we are going deeply further into this effort again, into this annotation. We recluster, we ensure, go to often much higher resolution, and then with majority voting, annotate these cells identities with particular labels. Now that in that process, yes, exactly. The harmonized labels are completely reevaluated, and for the human lung cell atlas, that resulted in 60% of cells being reannotated, often to a higher level of resolution, and sometimes just to a different clade in a hierarchy that we had built.
Evan, do you want to add? I also have a question that is similar on theme that you can answer to. Anything you'd like to add?
Yeah, it's really, I think, the second step that Malte was talking about in terms of once you start to get to more highly granular cell annotations, right? This is where there might be actually at the end of the ontology where you start running into problems. And then there really is a question of how do you actually start establishing these newer cell types in such a way that it's consistent throughout the community. At the moment, I think there is this expectation that the cell ontology will be improved, and then in the future, maybe one day we'll go back to these. But I think the original sin here is this idea that there's a single label, even if it's associated with the cell ontology, with the data sets, right? It's without the context you need to actually fit this within the cell ontology in a way that's useful. So I think taking the free text cell labels and then associating with the cell ontology, it was very useful for like standardizing the data sets for data curation, but in terms of doing the next steps of how does this actually fit into a human cell atlas, it lacks a bit.
Okay, I will thank you both for this answer. I'll follow up with a question and some thoughts to frame the discussion. Also from an anonymous attendee for CAP, how do you choose a reference to map new cell labels to? Is that organized as a hierarchy? So here referring to perhaps cell ontology that you just mentioned. And also, how do you deal with different granularity across data sets? And here I want to, bring up the fact that one path forward, which is bridging between Malte and Evan's presentation here, is that as we start assembling these integrated atlases from well-curated data sets, we're going to start having some gold standard data sets on which we can work towards consensus annotation, and that can become really great data sets in the future to start doing these label transfer. Just to kind of, frame the discussion here. So with that question, anything else, Evan or Malte, you wanted to add about the ontology?
I can try to answer that a bit. So with the annotation transfer methods, which I think are probably the most useful methods that we have at the moment, what people are used to, within CAP, there's really two ways you would be able to do this. One is that you should be able to use any data set on CAP as a reference, and then use that to transfer data set labels. The issue with this is that most of the really highly accurate annotation transfer algorithms, they require a lot of training on the reference, which means we have to process this data in some way. So working with this with a data portal is a bit tricky. So one of these methods actually is PopV. I think we have a computational solution though. So it's right that my goal would be that this annotation, labeled transfer should exist with all data sets on CAP. What's more likely is that for these really computationally expensive methods, we will look for these gold standard references that Chloe's discussing. I didn't quite understand the hierarchy sort of issue, but I think in terms of the precise or broad cell type labels, and this is something in terms of having tried to do this now for several months with people like David Sidney Sutherland, we want to actually ask this to people as opposed to, just mapping it back to the cell ontology doesn't quite show you within the data set itself whether this is a very precise label or a broad label. So I think of this as another metadata term that we could start calculating and asking users to give. And this should help us actually start standardizing these events.
Thank you. Malte, I have a question for you, which I'm going to summarize also from an anonymous attendee, asking you your experience about leveraging raw FASTQ files versus gene expression matrices, given the fact that people are perhaps using different parameters, alignment, frameworks. What has been your experience so far? Do you have any recommendation?
Yeah, I just saw that question as well, and I was smiling because it's very relevant to what we're trying at the moment. So I should say first that the HCA integration team is exists since September. So we're not all the way through to being able to fully answer all of these questions. That being said, we're trying. So in one of the projects, specifically for kidney, we're trying to remap every data set, realign every data set to see if that improves our integration. We have previously tried integrating kidney data sets, which is very challenging, given quite strong technical differences there. We are yet to see whether this realignment solves at least some of those. So I'm not able to answer that just now. What I can say is that what we're trying to, given the timelines also with which we were trying to generate these atlases, we are attempting mostly to go for count data, mapping between, so otherwise mapping on the kind of ensemble or gene name level, ensemble ID or gene name level between these references and between these individual data sets to ensure we have a consistent feature space. That has not failed yet. I will happily let you know when that does.
Thank you. We have another question. I want to make sure we ask, because that question was posted from the beginning from Sujash. For various tissue type, do you see a requirement for higher cell number samples for new cell type identifications? Also, what is the suggested read depth for cell for different tissue types to achieve a relevant gene expression profile? So that question could almost go to both, but Malte, I'll let you go first because you've been dealing with data sets of different size for the integration.
Yeah, I think this is, I can answer it only to a certain extent. This is not entirely my expertise. I think also something I would relate to, Chloe, potentially you as a data generator, you probably have other experience with this. We have set up a data set selection criteria for, as one of my bullet points of the challenges we're trying to address for integrated, Belting integrated reference atlases. And one of those is read depth. We have used that so far to filter out strong outlier data sets, which have very comparatively low read depth, which we know if it's not comparable to other data sets, this becomes more of a challenge because that is represented as a batch effect, which is more challenging to work with. But I cannot give you a cutoff to say this is good enough, this is not good enough. There are some really great papers on this, which I have no connection to. Otherwise, talking about kind of one read per gene per cell. I think there was, I read it, I'm not exactly sure who was involved anymore, but I'm pretty sure if you Google one read per gene per cell, you'll find it.
It's a complex question, deciding if you start with many data sets of all sort of size, which has some implication for batch corrections, versus starting with more sizable data sets in terms of number of cells and donors as your foundations, and then build upon it. There's no right or wrong answer, because smaller data sets maybe bring some very important diversity from a biological standpoint. So it's an excellent question, one for which we don't really have just yet a straight answer. Evan, I have a question for you. Do you have any solutions for unknown clusters in CAP? Do you help researchers to annotate unknown cluster? What about markers expressed in the same cluster? Can I annotate the cluster with two cell names such as ATL or TAL? I'm guessing that the gene names are very similar.
Right. So within the UI, you actually, the publication I was showing, in order to make a publication, you have to have the cell types annotated, so you can show this. If you own the data set, you can annotate the cell types, via selection on the fly. That said, the clustering doesn't need to exist in the data set. Clustering is sort of computationally expensive, and so we don't have a way currently with CAP that you could cluster on the fly. If you give me a second, I'll try to pull it up actually how you would annotate on the fly as well.
We may not have time, Evan. There's a lot of questions, but we can invite them to reach out to you for a live demo. So yeah, so there's an option to annotate live on the fly and to give several justifications, including several gene names, right? Mm-hmm. Wonderful. Malte, a question for you. In the single cell and single nuclear integration that you're running, are you running an algorithm to remove ambient RNA contamination? Is that something you run all the time or on a case-by-case basis?
I can give a quick answer, very case by case. This is something that, there is now also algorithms that do this without requiring empty droplets. But we typically in our hands, empty droplets based SUPEX, for example, outperformed non-empty droplet based SUPEX. And we often don't get full empty droplet data. for these efforts. And so we assume typically that the data sets we integrate have been pre-processed, have been QC'd and pre-processed. We do additional QC, but we typically have not been rerunning ambient RNA correction every single time unless this is specifically required.
Thank you. And Evan, do you have any rules regarding incorrect submissions? If someone annotates a cell type with a clearly wrong markers, how is that curated?
So this was, yeah, I get this question actually quite a lot. So we do have some basic checks to make sure that people aren't just uploading data sets, which are, you know, I'm waiting for someone to actually try to track us a bit here. So, but, you know, there is basic QC that we have. That said, this is part of the larger sort of data discussion with the HCA, right? So there's sort of rules that you'd have in terms of what should be the data sets associated with the HCA. And if at that step, people are sort of annotating cells with, you know, completely wrong annotations just to somehow trick up the system, that exists, that workflow already exists now. I mean, there's a level of trust here within the scientific community, right? That people aren't just sort of within bionetworks somehow annotating cells with, you know, false annotations. So that's the idea that there's people are actually being dishonest. In terms of incorrect, yeah, I think there's an assumption that people are doing their best to annotate these cells. At a certain point, if you do aggregate enough data, you should be able to, for previously uploaded data sets, give some sense whether those annotations are accurate or not, right? So that's part of this iterative process. Of course, that's the assumption that the training data that people have already given us is accurate. So,
but if I may jump in your user interface, you're building up some features where we can give some feedback if there's some disagreement with a cell identity, where you can actually give some form of justification of why you agree or disagree. That's something you're working.
Exactly. Well, that's part of the evidence. So that's sort of, there's free text that you would be able to say, you know, I disagree or agree with this. But there's also basic checks such as, you know, this is another reason actually why we want to have people give marker genes, right? So if people are sort of, if it's clear that the metadata associated with these cell annotations is completely off, right, and they're making a mistake, it'd be useful to flag this, right, and tell the scientists and the community.
So maybe if I could jump in,
yeah,
I think this is something where the collaboration between our projects also will greatly benefit, because we are re-annotating, and we have found also that actually based on marker genes that are reported, sometimes, you know, the annotations just don't work together. But I think in the long run, that's definitely something that will work well together.
Excellent.
Definitely. Malte, question for you. Can you speak more to the risk of overcorrection during integration? How can we check if relevant patient differences are being removed?
Yeah, that's a great question. And I don't want to get too long into this because I could talk about this for a very long time. There's, I think, the first part, I think you're asking about patient differences. I think the first question is, what is the scope or what is the goal of your atlas? Do you want to capture patient differences or cellular variation, right? And you can try to do both. It sometimes gets more challenging because at some point, you know, imagine two patients with very different disease phenotypes. Well, the difference is disease. Even the individual cellular phenotypes will separate out. So typically, we compromise a little bit on representing patient differences in the embedding, but then use the original gene expression data to model those patient differences where the batch effects are still in there and try to account for them separately. Overcorrection has different facets when looking at cellular, like cellular diversity. So something we found in the human lung cell atlas, for example, I'm giving that example all the time because that's, you know, it's done. And so a lot of these other ones are still, we're yet to find examples there. But so for the first version of the human lung cell atlas, we found that, for example, rare cell types that were pre-annotated, actually, tuft cells, ionocytes, were not easily recapturable by clustering in an individual cluster, depending on the integration method. So some integration methods just kind of merged a lot of cells into one central cluster, and then you could not, like rare cells, and you could not kind of recluster those out. So this is the danger of, specifically for rare cells, this overintegration. Other aspects that are not directly patient linked, but are kind of patient group linked is, let's say, disease. I'm sure if we integrate, so when we have cases, like the difficulty with integrated on the, for like capturing effects that are on the kind of super patient level or super donor level, is that their batch and bio effects, or like batch effects and these biologically meaningful effects, are typically confounded. So we remove batch effects, biological effects co-vary with this, we'll probably remove this. And so there we might be merging disease-specific cell states with their more healthy counterparts, not capturing these as well separately. This is something which, we're really trying to see how to improve in the blood cell integration with COVID and healthy cell states. And ways to combat that is like specific seed integration for disease-associated cell states to make sure when you benchmark, you capture these disease-associated states still in individual clusters. That's one way to do that. And also, of course, by reannotation. I've talked way too long about that already.
No, this is a hard question, and I have actually a question on the same theme for Evan as a transition. Evan, are you assuming that markers are the same for healthy and disease states? Most of the markers for healthy states can be lowly expressed in disease. Shall we think about markers for healthy and other disease? So, should we have different type of annotation? It's a hard question, but it kind of links to the question that Malte just had.
Yes, I think that is the short answer. There's going to be different markers between healthy and diseased tissue. I mean, the goal of this whole project really is that we would focus on healthy tissue first and then actually have some sort of atlas for the healthy tissues before we can start looking at the abnormalities of disease. I know that's the good stuff people want, but we need to do the healthy part first. In terms of CAP, it's true. I mean, this is part of the metadata. This is why we are collecting all other metadata associated with this data set. So if you know that this is diseased samples and they, annotate clusters of disease cells with particular types of markers, it's obviously going to be collected and used in as training data in a different way than healthy cells. So, and
I think it's important, the point you made earlier in your presentation about everything is going to be time-stamped because the annotation will evolve as we add more and more information. So it's conceivable that there's a cell state we believe is associated with disease, one particular disease, and with time we may realize that it's associated with several other forms of perturbation. So this annotation will evolve with time. But you are developing some user interface that will enable gathering that metadata, so we can refine this with time. Malte, I'm alternating between both of you if you hadn't noticed. So Malte, question for you: could you tell us about one matrix you use to compare data integration methods, how to judge if batch correction does not affect biological signals? And I'm saying you're currently typing an answer, but you want to give a quick highlight?
I can also do it like this. Sure. So actually, we developed 14 different metrics for different aspects of batch correction and biological variance conservation. Not all of these will be great in the same way, but across the board, we kind of see that they capture different aspects of this. For batch correction, for example, what we're looking at is per cell type, how do batches mix, and this can be evaluated by the silhouette width on average across batches, and that silhouette width should be 0 if batches mix entirely within a cell type. There are other aspects that have been published before already, for example, with the Harmony paper, this local inverse Simpson's index, which looks at what the neighborhood of each cell is, and if the neighborhood of each cell captures all different batches that should be there, basically. This is something that is, and then there's like multiple versions of this for cell type conservation as well, if those are like cell type labels, to make sure that all same cell types are neighboring one another or batches. And I can go on. There are many methods. There are ones that we have not used yet, which have been proposed recently, which maybe we should. And there are new methods we are adding, metrics we are adding. So if you have any other more suggestions, please shoot me an email. I'll take a look.
Thank you. Evan, for you, from David McKellar, in building a dataset for CAP, how can I incorporate other lab data? Will there be a process analogous to forking or pull requests on GitHub?
So that was the original plan. And this is part of the question that I'm now asking people within the community, is whether you're okay with that. So there's nothing like this which exists within the scientific community at the moment, right? I have my data. There's some sort of way that I've shared my data within some data portal, and anyone can go to that data set and basically take a copy of it and make corrections, right? So I'm happy to have that model, and I think that would be quite interesting. I'm not sure if others would really. I still don't have a good sense of whether it's that's the best way for this to work. So, but it's certainly technically possible, right? I think it's mostly just we need some more buy-in from the community, whether that would be useful.
Thank you, Evan. Yeah, definitely. This if
I should say also,
yeah, go
there
ahead,
is a way
please.
now within the community that you can, I'm sorry to interrupt you, Chloé, but
No.
there's a way now within CAP that you can basically ask to have permission to view the data set. That's a bit different than a pull request though, right? A pull request is you can, or a fork, right? A fork is there's no permission. So that's the difference. Sorry. Continue.
No, that's an important point to add. Malte, an important fundamental question for you. What is the difference between batch correction and data integration? Because you've been using both terms to make sure that the audience understand.
I use, so, okay, I have kind of my own little distinction between these two things. This is definitely not shared in the community. I have written about this previously. So technically, a lot of people use these two terms interchangeably. In my mind, I think about batch correction as in different complexities, or data integration different complexities. When you have multiple samples from the same data set, where you expect compositions and to be very similar, where simple methods, even like global methods like combat, can work well. This is, in my mind, a batch correction problem. Data integration being across more nested batch effects, where data sets can differ in compositions, technologies, and there's a lot more differences to overcome, and typically different methods are appropriate for that setting, which I call data integration. I have used those two terms interchangeably, however, as is generally used in the field. So yeah, that's not particularly helpful. Sorry.
No, that was definitely helpful. I want to read a question along with an answer from David Osumi Sutherland, who actually is leading a lot of our ontology effort. So Shangshu Yao wrote in CAP, do you have a set of control vocabulary ontology for cell label ready, and can it be shared? And David answered for us, CAP supports the cell ontology, but also free text cell annotation. The cell ontology doesn't always have appropriately granular cell types or synonyms that allow cell types to find with text search. We plan to iteratively use annotation on CAP and link references along with the outputs of the Jamboree to extend and deepen the cell ontology with new cell types and synonyms. And so David is, so thank you, David, for this answer. David is one of the cell annotation platform investigator. We're almost at time. I want two quick questions to wrap up. Malte, what is the expected output for the integration effort? Are we expecting H5AD files with some harmonized metadata and normalized counts?
That's I mean, I think a lot more than that, I would really hope. So there's, I think, the expected output comes also along the lines of like what use cases we find along the way, right? So something which I envision kind of very clearly in my head is to have reference atlases that are useful for consensus cell type annotation, right? To find a consensus within the different bionetwork communities, and then also across the bionetwork communities in the case of bionetworks like immune, right, which have input from everywhere. So that's the first aspect. I think it's this consensus finding tool, which can then move towards kind of CAP for more decentralized approaches for this and iterative improvements of this as well. The second aspect is rapid like facilitation of like very rapid data analysis. In my mind and in previous kind of approaches, little smaller projects, we found that actually using a reference-based analysis approach drastically speeds up the time which we need to analyze new data sets. So this is something which, you know, within a couple of hours, we suddenly have an annotated, decently annotated new lung data set by mapping it to the human lung cell atlas. And there we're looking to kind of see different portals for this. Where that in event lives is still, I think, up for some debate. But several different kind of approaches to map your data to something which can rapidly help you analyze that, that being a second output. And then, you know, there's a lot more I could go on for different use cases that we will find and still hope and have found so far. But I think Ellen wants to continue.
To wrap up, so I'll just some concluding words quickly about where the data will live. You're going to hear more in the months to come about the central gateway where we'll be hosting some of these data in Portal. Quickly answering the reference atlas regarding liver. Of course, this is part of the Human Cell Atlas. I think Malte is answering this question. And then for cell ontology, will labeling cells with a standard code just like gene is a good idea? I think this is a more complex question that we're happy to follow up upon. And so please reach out to Evan and I for more questions. So thank you all for all your questions, for joining us for this seminar. Both Malte and Evan are very eager to connect with all of you to try to work together as a community to build this Human Cell Atlas.
Thanks, everyone. Thank you.
Thank you.
Goodbye.
Thanks, everyone.